It's a little bit of pressure to be the last talk, keeping everyone here, but my name is Evan Brenner. I'm a postdoc, also in Janani Ravi's lab at the University of Colorado. And my questions are, why do pathogens infect the hosts that they do? So I'm going to start off with what is hopefully an easy sell, and that is that infectious diseases are bad. Here are some of the emerging infectious diseases in just the last 2 decades. It's a lot. And worse, the rate of emergence for these infectious diseases has been accelerating over time. As a bench-side biologist during my PhD, my research interests were in these zoonotic infectious diseases. And a zoonotic disease is one that transmits between animals and humans and back and forth. Most emerging infectious diseases are zoonotic in origin. Zoonotic infections include many on the map from the last slide, including COVID-19, which you may have heard about. All of this occurs in the One Health context, which is a framework that reminds us how human health, animal health, and environmental health are all interconnected. And if we want to really solve any of these problems, we need to consider this larger context. In that framework, the problems that are driving infectious disease emergence are complicated. Climate change, deforestation, urban expansion, animal agriculture, all of these push ecological niches to overlap where they didn't before, and that risks microbial spillover between these niches. But not every microbe poses a threat. So how do we rank the risk that each of these microbes might pose? And for pathogens, which are the molecular mechanisms that really drive this risk level? Towards these questions, I believe that microbial genetics will help explain a microbe's adaptation to its host or hosts of origin. As you all know, we have vast quantities of publicly available microbial genomics data, but how do we use it effectively to answer some of these questions? And that's where Dr. Janani Ravi comes into the picture here. I joined the J. Ravi lab last year, and I started learning how I could use interpretable supervised machine learning to associate microbial phenotypes with genotypes, much like Ethan. But genotypes contain many layers of information across molecular scales, right? So where do we focus? We know that changes in gene content do play roles in host adaptation, and gene counts are still relatively manageable in terms of ML feature space. They're also among the easiest for biological interpretation, but I notice I did not say easy, among the easiest. But nucleotide k-mers can span actually a few of these scales at once. While they're less immediately interpretable, you can map k-mers to genes, and you can capture variation in intergenic regions as well. Plus, k-mers can capture polymorphisms, which can be helpful for host adaptation questions. So which am I choosing? It's both, actually, but I'm starting with k-mers. I want to use machine learning to associate pathogen k-mers with the phenotypes of the host that they came from. So to demonstrate this, I've taken many genomes from 2 different zoonotic bacterial pathogens. Staphylococcus aureus is a major human pathogen, hundreds of thousands of deaths per year, but this is also infectious to agricultural and companion animals as well. And Streptococcus suis. It's a huge problem for the swine industry worldwide, but Strep suis also spills into humans and can cause serious disease. And fun fact, most of the Strep suis strain typing in the world actually happened here at the reference laboratory in Montreal. For these 2 pathogens, I extract DNA k-mers as features from their genomes. And to perform the machine learning, I'm using a set covering machine implementation called Cover, developed also right here in Quebec. This is a coincidence. I didn't plan all this. That QR code there is actually a Google Tech Talk by the then graduate student developer of Covered, explaining the implementation and some really cool tricks that they did to handle processing these enormous k-mer matrices. And I thought it was a pretty fun watch, so check it out. But later, I'm still going. In the short version, our machine learning features are a binary k-mer presence-absence matrix spanning all the isolates in a species that we're considering. And our labels are just the hosts of origin for each isolate. For Staph aureus, they're grouped more broadly into human versus non-human. And the goal is to identify as few k-mer rules as possible that still best predict the labels. And for Staph aureus, how does it perform? Quite well, actually. For 720 Staph aureus genomes and at a k-mer value of 72, we strike a pretty good balance of performance metrics and compute time. Now, which k-mers are actually giving it this good predictive value, though? Do they actually make biological sense, or am I fitting spaghetti to the wall? In this set, the top scoring k-mer maps here by this dark red indicator it maps to the phage integration region of phage phi SA3. So it's a hit on a horizontal gene transfer element. And it turns out this phage brings with it a set of 3 genes, the human-specific immune evasion cluster. It's characterized in the literature to be involved with human adaptation and hypervirulence. And not only that, when clades bearing phi SA3 spill back into animals like livestock, they actually tend to lose this phage. So this really does look like a strong signal of host specialization. And for strep suis, I used 1,500 genomes. I looked at swine versus non-swine origins, and it's still performant. And of 3 k-mers identified, the top 2 of them map within the capsular polysaccharide synthesis locus, specifically in insertion sequence elements. This region appears to be a pretty frequent target for mobile genetic elements, which makes a lot of sense because this is also associated with serotype switching. And it's a locus that the folks at the reference lab down the road probably know pretty well based on that. The literature also supports this locus as deterministic for mediating host cell interactions. And potentially this could be another hit for host adaptation then. The 3rd k-mer is also in a different mobile genetic element. Are we sensing a trend? Yes, me too. And going back to some of the earlier talks today, how many roles is horizontal gene transfer really playing in adaptation of all of these different phenotypes? It's very interesting. The fingerprints just keep popping up in like every phenotypic crime scene that I look at. So backing up, I'm happy to report that this project that I'm spinning up and going to pitch as an NIH F32 is pretty fun. I like it, but it's not perfect. For one thing, metadata availability, completeness, and reliability is at best kind of poor. This limits both the questions that I can ask and the confidence in interpretation for my results. And host of origin does not require that the host was sick. This approach is giving me good signals of adaptation so far, but about 25% of you in this room are carrying Staph aureus right now in your skin or your mucosal flora, and you're healthy as far as I can tell. So presence does not strictly mean pathogen. Finally, models are, by necessity, built from data of the past. So how are they going to deal with new diseases and new features that arise in the future? That is a bigger question. I'm still wrapping my head around that. But on that path, I want to work with collaborators who already have good curated data sets for their organisms of interest, because if I can really trust the data and the metadata, then teasing out these biologically relevant insights seems very manageable, and validation becomes more straightforward. But I do want to test the edge cases, and public data sets offer plenty of edge cases, don't they? If I can still get useful features predicted out of these noisier data, like what I've shown in this talk, then I think that's also a win. And because a lot of emerging infections are in fact viral, I want to play with some viruses on the computer. This k-mer approach worked great for smaller viral genomes where single base changes can have huge impacts. And I've actually set up a simple model for influenza A using very short amino acid k-mers. And it works really well to predict host, but we are short on time. So ask me about that later. A huge thanks to Janani and her whole lab group. Thanks to ISMB for letting me share my story, and EvolCompGen for helping cover part of my registration. And of course, thanks to all of you for listening. I would be happy to answer any questions in the minute we have left.
Thank you for the really clear presentation. So when you're doing your machine learning, did you explain where do you get your training sets and whether you have to train it kind of separately for each pathogen?
Yes, sorry, I didn't go into the details because I didn't go into the details. I didn't have a lot of time, and I prioritized the biology because I am a biologist by trade. But to answer that, the data that I'm actually acquiring are from 2 sources. For the first set for Staph aureus, this is pulled from NCBI. They're just annotated as complete genomes, so I don't have to worry about draft quality. So that's just NCBI and then formatted through DSK, k-mer counting for input into cover. For strep suis, that was actually taken from a new ENA repository called All the Bacteria, which is a preprint. It's kind of cool. Go check it out. And then, I'm sorry, what was the second part of the question?
Whether you need to train it separately.
Oh, yes, absolutely, yes. Yes,
Yeah.
for each individual species right now, I am training them separately. And depending on the species that I'm analyzing, like even if I just stick to k-mers, I'm expecting the k-mer length to be quite variable, which is optimal for each species. So I do want to look at other features and see what I can do.
Well, thank you very much. I just want to have a little shout out also to Janani, who couldn't join because of visa issues, but who has been really very involved in the organization. So well, we miss you, Janani, and as well as all the other remote attendees who couldn't attend because of visa issues. We are aware of this. Is really been pretty bad this year. So thank you.
Thank you.
Well, and we're now at the end of the session for the day. So, any questions to speakers, you can do one-on-one.
Catch me in the hallway.